one of the big deals that's happened in science in the last half year, really, 6 years, 5 or 6 years, is the continued better understanding of genes and DNA and how it might be modified for therapeutic purposes. And George Yancopoulos has a history of understanding things pretty fast and developing technology in line with the latest things that are happening in science. His background is Columbia University, where he was a medical student, got an MD, PhD, and was a spectacular product of Columbia, then went on almost immediately to join Regeneron Pharmaceuticals, where he was essentially the founder of the science group, has recruited the whole team, and has generated much of the technology that is there and has been behind every drug that's come out of Regeneron. He is a source of all the ideas that I ever have in science because I talk with him, and he actually teaches me what I should know about the developing sciences. And there's no one who understands it better than he, and so I'm delighted that he's here to discuss this topic, because it's a quickly breaking topic that's going to be very important in the future. George Yancopoulos.
Thank you, Roy. I'm going to take moderator's prerogative a little bit and go backwards before we go forwards to the panel and get to the specific topic of discussion here, which is the molecular scissors. I want to go backwards to a couple of the previous panels and also the last talk by Mark, which I thought was really a great talk and highlight a lot of issues. But what I see us as scientists or as specialists, that we very much get into the details, and we even suggest these tactical solutions and so forth, which I'm not saying are unimportant, but sometimes we really lose the big picture and what should be the really big message here. I hear about healthcare costs, and I hear about efforts to cut them down and so forth. But you know what my big take-home message from Mark's talk was? Did you guys notice in one of his charts that 40% of the deaths are obesity-related? Okay? Do you know 50% of Americans are obese today? Less than 1% were in 1974. Okay, another big take-home message. Looking at the life expectancy charts. In 20 years, more than a third of the U.S. population will be over the age of 85. More than a third of us will be over 85. And among other things, that means that half of those people, just in that age group, 1/6 of the U.S. population will have Alzheimer's disease. These numbers are staggering. You talk about freaking doomsday scenarios. Okay, you got climate change and global warming on one hand, and on the other hand, you have the very survival of our species. We cannot lose sight of this, guys. We are at freaking war. Okay, there is a war for survival of our species and survival of our planet. Who is going to save us? Okay, maybe we can do some social behavior changes, and affect change a little bit in that way and so forth. But guys, you know what we're counting on? There's only one thing that can save us. It's science and it's innovation. And there's nothing more important than that. And we have to take back the conversation. Okay, we've lost sight of that. Okay, there's a lot of things this industry and we have to apologize for, but the big picture is, We're the only ones who are going to be able to save the world. I'm not talking about scientists only in healthcare and biology and so forth. It's overall. It's a war. It's a war of survival. And if we don't realize it, okay, we're doomed. And this has to start, we have to realize you get what you pay for. There's nothing more important than serious, long-term investment in trying to save our species and our world. It starts at the earliest ages. Okay, it starts with education. It starts with promoting the best and the brightest minds to get them into the most important area they should be getting into, which is the sciences. And it continues on. If we don't change the equation, if we don't change the conversation, if we don't communicate to the world that the most important thing, talk about value, the most important thing that we can do is invest in science and in innovation, and otherwise it's all over. Okay, and if we don't start doing it now, it'll be too late. And along those lines, people have to realize also that, for example, I don't know, to cure Alzheimer's disease, you don't invest in doctors who are working on Alzheimer's disease. You have to do really fundamental scientific work. And the real answer might come from a fruit fly or from, signs in a worm. And now I want to get back to today's panel. Because it's a perfect example of what I'm talking about. What we're talking about today is truly, potentially, a new transformative field in science. Something that actually can make a difference, that can save lives, that can change the equation. Okay? And where did the fundamental discoveries come from? Okay, they didn't come from trying to cure a disease initially. The fundamental discoveries came from actually looking at some bizarre immune defense mechanism system in bacteria against bacteriophage. Okay, and now that's leading to potential cures for human diseases. So when I talk about investing as a society in science and knowledge, I'm talking about real deep fundamental investment. I think that's what we really have to do, and that's what got us here in terms of this panel. But before I turn it over to the panel so that they can talk about what they're working on now, I think another really important understanding that we have, I think this could be the dawn of a new era in terms of the approaches that we're going to hear about today. But we also have to understand that we can't overpromise or not appreciate the investment and the time that occurs. So, for example, it's possible. Do you know that there is a protective gene change for Alzheimer's disease? Which means that you could, if you could have everybody in the world have that mutation, there'd be X-Men. They'd be protected against Alzheimer's disease. That problem could be solved. It could be solved by this technology, but not by this technology today. At current rates, there is no way to gene edit a gene in the brain. But it could happen, okay? But it's going to take a long time. It's going to take a lot more investment. But that's someplace where we could go. Gene editing the brain to protect against Alzheimer's could cure an epidemic that we have no solution for today. But we have to understand, you have to start small. Right now, gene editing is not an immediate panacea. You got to walk before you can run. There's only some tissues in the body that you can even get even a little bit of access to, and make a little bit of a difference to. And so the companies and their representatives that we're hearing about today are some of the pioneers in a field that could be making a huge change, that could lead to a true revolution in decades from now, but it has to start somewhere. And these companies are the pioneers that are making the first attempts to take this brand new science and this brand new capability into patients. So what I'm going to do is I'm going to ask each of them in turn to introduce themselves and spend about 5 minutes talking about the efforts of their companies. And then for the last half hour or so, we'll open it up for discussion with the audience. So we have with us, as I said, representatives of 3 of the leading companies in the field of gene editing. Charles Albright, who's the recently appointed Chief Scientific Officer of Editas, which is one of the premier companies now in this CRISPR space. We have Sandy Macrae, who's the President and CEO of Sangamo BioSciences, which is using a totally different technology having to do with zinc finger proteins, but also allows you to edit genes in a variety of ways. And he'll tell us a little bit about their efforts. And then you'll also hear from John Leonard, who's the Chief Medical Officer of Intellia, another company that's leading in the CRISPR field. In terms of full disclosure, I should just say that we do have a major collaboration with Intellia and with John, but I think that all of these companies are really doing pioneering efforts and setting the stage for what could be an entirely new world and a new future sometime from now. So with that, I'll ask Charles to introduce himself.
Hi, I'm Charlie Albright. I'm from Editas. It's a pleasure to be here and a pleasure to speak first and a daunting challenge after that introduction. So at Editas, we're trying to use CRISPR-based gene editing to develop transformational therapies. And unlike a lot of approaches to drug discovery, this one has a significant technology component to it. So about half the company is devoted to basically industrializing the technology. And so there are many parallels between small molecules, biologics, and CRISPR in the sense that you have lead seeking and lead optimization, but the actual mechanics of doing that are radically different because you're using a nucleic acid-based approach here because you have an RNA-directed endonuclease for DNA. And so by definition, you have genotoxicity risk. And so you're cutting DNA, and you want to be sure that you're cutting it in the right location. You're not cutting in the wrong location. In addition to miscutting at individual sites, you have the potential for translocations and larger genomic rearrangements. So a lot of the technology that goes into actually figuring out whether you did what you wanted to do, the specificity assays, if you will, of drug discovery are being developed in the same time the therapies are being developed. So a significant chunk of our company is developed to making that technology base real so that we can identify guides that do not have off-target cutting. There's a limit to that, to the sensitivity of that based on today's analytical methods, but we can get down to no observable cutting at the 1 in 1,000 off-target rate. So it's really quite impressive when you optimize the guides. I can say that people who do this in their garage over the weekends don't have quite that level of success, and that's one of our concerns, quite frankly, in the field, that the technology is actually quite amenable to the typical molecular biologist. The other half of the company is trying to take that technology and turn it into drugs. And so, as George alluded to right now, one of the huge challenges with these modalities is actually delivering them. There are 3 tissues where we can deliver to easily today. That's ex vivo, where we take cells out of the body, do something to them, put them back in. There's the eye, where local adeno-associated virus has been shown with gene therapy to be quite effective. And there's the liver, where you can go with either lipid nanoparticles or viral delivery as well. So that's where most of us, I think, are focusing now. We have programs at Editas. Our lead programs are an inherited retinal disease, a relatively ultra-rare disease, but quite severe, leading to blindness. We have a collaboration in CAR T cells with Juno. The notion of editing the CAR T cells to overcome some of the immuno-oncology checkpoints and tumor microenvironment suppressive mechanisms that dominate the effectiveness of those molecules is quite exciting. And we have interest in targets in the liver as well. So it's very early days, but the ability to reduce these technologies to drugs that can go into people is happening very quickly. We've seen academics take that approach already with CRISPR. We hope to be in the clinic in the next year to 18 months, and these could happen pretty quickly. So it's an exciting time from that angle. It's also an exciting time from the public policy angle because in addition to the problems that George mentioned, there are issues with editing embryos, gene drives, all kinds of policy issues that one does not typically encounter in an early technology drug discovery forum. So I think with that, I'm going to pass it to my to my colleague or back to the moderator to pass it to my colleague?
Sandy?
Good afternoon. It's a pleasure to be here as part of the 3 people that are going to save the world. So I joined Sangamo 4 months ago as the CEO, and it's a remarkable company. It's one of the pioneers of gene editing. It's been around for 20 years. And it uses a different technology from CRISPR. It uses protein transcription factors that hold on, the fingers hold on to the DNA, and then cut it. And in that cut, you can either disrupt the gene, you can insert a gene, or you can alter the transcription. And it truly is a technology that we have worked on over 20 years and have a control over that is beautiful. It is science. It's almost at a level of engineering. And what I've done with the company over the past 4 months is we've focused on what we're going to deliver. And we've opened our first clinical trial in gene editing, but we've had patients treated where we've altered their T cells in HIV, over 70 patients treated with gene editing already. But there's 4 things that we're going to have in the clinic at the beginning of next year. 3 of them are gene editing. And in these, what we do is we edit, we cut with the scissors one of the introns in the albumin gene in the liver, and we drop in a transgene. And that transgene is then expressed through the albumin locus and creates the protein replacement that the patient's got missing. So we've opened a trial for haemophilia B, and that one's recruiting at the moment. But next year we'll also add trials in Hunter's and Hurler's syndrome. So for each of those patients, we will give them a permanent solution to their disease where they'll have their enzyme replacement from the moment that we insert the gene in the liver. And I think this is a fundamental moment in gene editing and gene therapy. It's the first time anyone's managed to do this. Behind that, we look at thalassemia, we look at the T cells like everyone else does. And we also have some very interesting results more recently in Fabry's disease. And even further behind that with the scientists, and they are remarkable scientists at Sangamo, they have a fantasy toy box of molecular biology, doing things that, as a molecular biologist, I could only have dreamt of 10, 15 years ago. Because my reflections on this, and I want to step back a bit from the technology, because I think in the end it's gene editing. And 10 years from now, we'll talk about gene editing. We won't talk about the different technologies. And 50 years from now, we'll just do this as a standard form of medicine. My reflections are severalfold. One is the technology is really advanced. The molecular biology, I would say ours is better, of course, but even CRISPR, the capabilities that we all have to edit is remarkable, and that's no longer what's limiting the application of the technology. We can now cut every base or its neighbour within the genome. We can do it at an 85% efficiency. We can do it without cutting anywhere else in the genome, or as far as we can detect anything else in the genome, because, as you said, we're now at the level of the assay detection. We can do it at clinical scale. We can do it and put it into patients, and it's reasonably safe. So now what's limiting in these technologies is where you put it and how you get it there, and which diseases you're targeting. I'll come back to that latter one later. To me, the limiting factor in gene editing is no longer the molecular biology, it's the delivery. As you said, we can put it into eyes, we can put it into livers, we can do it ex vivo. The moment that someone discovers a reliable way to put it in other tissues, this field will blossom, and the number of genes that will be edited will increase logarithmically. One of the things I discovered when I took this group on is they can edit tau. They can control the expression of tau. I did ask them how they were going to use this, how they were going to apply it. And they're doing it on the assumption that eventually we'll be able to put this gene editing within the brain. And one can only imagine the experiments you'll be able to do once that is possible. So delivery is the key to this. And I truly believe that delivery will be solved in the near future. And I truly believe that CNS delivery of these gene editing tools will be solved in my lifetime. And then the second thing I want to talk about is how you compare these different technologies. Because just like we all got used to thinking about efficacy and side effect for small molecules, so as we can all really understand what's a good drug and what's not a good drug, we need to get used to having a language that describes how you use gene editing. What does it mean about 85% versus 15% cutting efficiency? What does off-target mean and what's important? And what kind of scale do you need to think about for this to be meaningful? It's quite easy, as you said, to do CRISPR as your PhD or your postdoc. It's a much different thing to convert that into a medicine. And we need to be careful that we're discussing the same thing as medicines. But my final kind of piece I wanted to reflect on comes back to the job I used to do. I used to be Global Medical Officer for Takeda. And as Chief Medical Officer, I used to think about how you gave medicines to patients and what was a benefit-risk balance. And I think even though the technology is racing forward, we need to think about and talk about some fundamental benefit-risk discussions. And yes, we can talk about gene drives and embryo manipulation, and those are big picture, almost societal discussions, but there's almost like a small medical discussion that needs to happen around gene editing. What's the benefit risk? Which diseases is it appropriate to treat with gene editing? Should you treat hypertension with gene editing? Should it be a disease that the patient's going to die for? Should it be in children? Should it be in adults? I think those are a series of discussions that we all need to have together. And then we need to think about safety. If I do edit in haemophilia, putting a transgene in someone's liver, it's there for life. And therefore, I don't know what safety looks like for life. I know it needs to be as clean as possible. I know it needs to edit in as few other places as possible. But I don't know how to have that discussion with the patient. If I have a discussion with one of you about an antihypertensive, I know that if you have a reaction to the drug, if you have a rash or your blood pressure drops, if I stop the drug, your blood pressure will come up and the rash will go away, almost always, all the time. If I edit something in your liver, it could be that you're left with that for life. We need to find a way to talk about that so as we can consent patients properly and have a proper discussion. And then that leads to the final thing about what's our responsibility once we do it to patients. Once I put this gene in the patient for factor 9 or for hunters or hurlers patients, they have it for life. And I feel I have an obligation to follow them for life. I have an obligation to make sure that this has got no consequences. Some of our competitors use lentivirus. And lentivirus, instead of being scissors, some people would describe it as a shotgun, where you blast in new copies of genes throughout the genome. And people have said, well, nothing has happened. Well, of course nothing has happened, because if something bad was going to happen, it wouldn't happen immediately. What will happen if any of us get our technologies wrong is that we will acquire mutations or we will lose control of oncogenes or controlling systems that will gradually separate from control over many tens and 20s years. And that's why we need to follow these patients for a very long time. So I think we can have this conversation about the technology, and I think that's an important one, and we will all believe we're right. But I think we're all agreed that this is a complicated application of the technology, and we need to start with the patient and think about how we use this carefully and prudently.
Thank you. So hello, I'm John Leonard, CMO of Intellia Therapeutics. It's a small company that began just in the last 2 years. The technology, CRISPR-Cas9, is new, and our company's even newer. We've gone from literally the 2 people that I was a part of to about 100 very, very quickly, I think reflecting the excitement, the incredible opportunity that we have. I'm not going to repeat the themes that I think are absolutely correct, and Sandy and I have sat in the same room and other places, and I think by and large you've done a very artful job of identifying issues that are absolutely front of center for gene editing. They are things that we're going to need to come to terms with. What we're trying to do at Intellia to do that, because we bear those issues in mind, is think very much along the lines of what the nature of the edit is and where we can get it. And I absolutely concur that there's 2 fundamental problems that we're always going to have to address. One is delivery. Can you get the technology to where you want to take it? And then what do you do when you get there, and how precise is that change? when we thought about delivery at Intellia, we've divided into two broad categories. After ex vivo and vivo, where you either take the cells, the technology, or go into the body, there's a chemical approach or a biological approach. And we have been excited about a chemical approach, or lipid nanoparticles, where we know we can get to very high levels of delivery in the liver, where many will know there's a very, very long list of diseases that emanate from there. So taking that notion, in fact, that you said, Sandy, which I think is correct, that once you get to a tissue, there's a lot to do there. We believe that we've cracked the code, if you will, at least in animals, with respect to the liver, and are now working on ways to extend the nature of the edit from a simple knockout or turning off a gene, if you will, to actually introducing small or even larger amounts of DNA. To do that, this notion of benefit-risk, I think, is absolutely essential for all the reasons that have been stated. These are, by design, usually permanent changes, and that merits very careful consideration of what the disease is that one is going after. So we tried to be very mindful of that, and with our ex vivo approaches, we think primarily of oncological problems, and this is taking lymphocytes and modifying how they'll behave, CAR-Ts and the like. But in the in vivo setting, where you've actually changed the tissue that's there, we've looked for conditions that we believe are very serious, mortal either in the short term or the long term, things like inborn errors of metabolism, genetically caused COPD, alpha-1 antitrypsin deficiency, and the like, and even hepatitis B, where 300 million people carry a virus, unwanted DNA, if you will, that can be destroyed by very specifically opening up that form of DNA that's there and having the body destroy it. So we're trying to take these two Venn diagrams of delivery and editing, bearing on top of that a view of what is an appropriate use of the technology, and starting our preclinical work. We're not in the clinic yet, and we acknowledge the, I think, standard setting that you've done, which has shown that gene editing can go into the clinic, and there is a regulatory pathway, but we're hoping to be in the clinic with you in the next 2 to 3 years.
All right. Once again, I'll take the moderator's prerogative and just ask you guys maybe the first question, but I firmly believe that you guys have in front of you a series of challenges and hurdles and landmines and so forth to get by, which you've described, in order to get your first sets of therapies, hopefully working in patients with a good benefit risk and getting approval and so forth. But as we've heard, most of these are targeted to these accessible tissues, the liver, the eye, or tissues that you can take out and manipulate, like T cells or stem cells and so forth. So I think that assuming that all does work out, looking at what you guys defined as the challenge, delivery, what are you guys either doing or what do you guys envision for that future that could really be transformative? Do you guys have programs that are imagining how you're going to take the delivery problem right now, which is how do you get better delivery to the eye, or better delivery to the liver, or better delivery there, and actually change it to the really transformative question, which is, can you deliver it, for example, into a single beta cell in your pancreas to cure type 1 diabetes, or can you get it magically to enough brain cells to protect against Alzheimer's or cure Parkinson's or do something like that? Do you guys have any programs you're working on to really address the almost science fiction possibility that you could do that? Let's start with whoever wants to take that first.
So there's one of the things I did when I took over the company was we looked at the research group, and they were almost entirely focused on the zinc finger platform. It's clear that we need to broaden and move into adjacencies such as vector delivery. So we are looking at both lipid nanoparticles, which I think is a great hope to get us away from viruses, and looking if we can create new, I never thought I'd say this, you could create new virus forms that aren't recognized or have different tropism. But there was even a publication I read this week where people were making use of retrograde viral flow. So in viruses such as rabies and polio, they can go retrograde within the neuron. And they had shown that if you inoculate, and they had created an AAV that you could inoculate certain regions of the brain and then cause that spread and show that it went to the regions in the brain that that tract went to. Once you can start to do that, there are so many pathways that we can influence. Things like Parkinson's disease, Huntington's disease, are tractable. Carefully, but are tractable.
I can take a shot at that. Following on the chemical approach, which we like because you can control it, and assuming less than 100% efficiency when you expose a patient to a treatment and you need to come back again, it's a viable way to overcome immunogenicity of what might be a biological approach. So we're very interested in finding ways to derivatize those lipid nanoparticles. There's interesting ways where you can think of adding moieties that will direct them to specific receptors that reside in very specific cell types. There's always going to be a physical-chemical limitation. They're a certain size. They have to be able to get there. But again, going back to this notion of benefit-risk, there are conditions where an injection into the brain, for example, would absolutely be appropriate. And we're looking at that now. We're looking at novel ways to, again, using chemical approaches to get both to the front of the eye and the back of the eye. And it's a lot to do. There's a lot of tissues to screen for. And we've sort of tiered them from more readily accessible to less. But the intention is absolutely to scan as efficiently as we can and look for additional technologies that can branch off of what we already have.
And we're doing effort as well at Editas. So we announced fairly recently that we have a collaboration with the Cystic Fibrosis Foundation, and a good deal of that collaboration is to look at the various delivery modalities that are out there. And we're looking at Duchenne's as well as skeletal muscle disease and ways to approach that. And so I think there's efforts obviously ongoing in our firms, but there's a tremendous effort in the field and driven mostly by the gene therapy companies. And so I've seen it in meetings. I think it's published, but at least in rodents, you can, for instance, systemically administer adeno-associated virus and get it broadly distributed in the CNS. So I haven't seen anybody reproduce that in a higher species yet, but there's clearly a lot of effort in that area to develop vectors that would access other parts of the body. And it's something that we, for the reasons that Sandy outlined, we're all paying really close attention to, because every time we can get to a new tissue, most of us have a mental list of the diseases we go after if we could access that.
All right, great. So I'll open it up to the audience.
So taking on the science fiction side, maybe we are at the brink of a true revolution in the history of mankind. We can think of the dawn of fire and the origin of agriculture, and maybe we are on the dawn of seeing Homo sapiens 2.0. And I wonder if that Homo sapiens 2.0 would be a Cas9 Homo sapiens transgenic. What do you guys think?
Cas9 transgenic, is that what you said? Where humans carry the enzyme? I do not see that in the foreseeable future. I mean, just to, it's so tempting, I think, to think really big and imagine generations of what the technology might be and where we wind up. I prefer to think of it in terms of problems that are solvable now. And I think the first issue is finding that set of somatic disorders where it makes sense to take the plunge, take the risk to actually go and make a change that, as we said, may be permanent. And I think it's going to be this stepwise accumulation of data that happens where, assuming we don't misstep, we gain increasing confidence that we can do this safely and that we can assure ourselves that there aren't off-target effects that play out in humans over time. And so as much as I'd like to think big, in terms of delivering all kinds of genetic therapies to large numbers of people at low cost, I mean, that's a future I don't expect to see. I hope I'm wrong. But, yeah, I see years of very hard, incremental work where we do something and we wait and we watch. And we do the problems that people can look at and say, that's the right thing to do, and we're comfortable about it. And because of that, a company like ours, and I think it's broadly true in the industry, just doesn't think about changing the human germline and taking those risks, not only for that generation, but for all subsequent ones.
Though I think it is worth mentioning, I do think that there's a lot of possibility for, in many ways, changing the human organism for the better, not necessarily in the germline, but just all the things that you guys brought up. And you have to understand, I totally agree with these guys. These guys have to do a lot of heavy lifting and work over the next few years to turn the first generation of this technology into reality. But the next generation, I mean, I think we are talking about improving people who are at high risk for Huntington's disease, like you brought up. You can, if the technology exists, you can go in there and you can take that susceptibility away from them. Or even a dream like we were talking about before. You can actually go into brain cells and protect them with benign mutations against the future risk of Alzheimer's disease. I mean, these are theoretically possible now. And I think those things are worth thinking about.
It's more than theoretically possible. We can do that with Huntington. So we have a partnership with Scharf where you can't take out the Huntington gene because it's necessary. But what we can do is we can turn down the mutant allele and the trinucleotide repeats and not touch the normal one.
Yeah, I believe very much, I agree, that the DNA side of it is more than theoretical, but it's still left to, getting into the right cells in the brain and doing it widespread enough. And that's what I'm talking about. But that's hopefully going to be the next generation after you guys, solve the first set of problems, which are obviously what you guys have to do first to make this a reality. But I think the future is incredibly exciting.
So I'd like to ask the panel, some of what I've heard reminds me eerily of what was discussed about 10, 15 years ago about stem cells. And there have been a number of setbacks in the stem cell field, mainly safety. So, and the same thing can be said, I think, about the gene therapy. There was a moratorium and so on. So what kind of safety measures need to be put in place to avoid the fate of stem cell therapy, which basically I don't really think has delivered anything of substance? And again, some people could say this is hype or whatever, because when one needs, and having personally been involved with the FDA in terms of the regulations, the problem is that the FDA has to play catch-up. Because when it comes to conventional drugs, the FDA know exactly what they want and what they don't want. The problem here is that they constantly have to play catch-up, and therefore they will say to you, well, if we do this, will you accept that? And they'll say, well, we'll let you know when you show it to us. That financially is just not feasible. We need to have pre-IND meetings and end of phase 2 and so on to craft a path to a drug, a therapy, or an intervention, whatever it is. And I think that in that regard, stem cells have failed. What can be done to prevent that from happening to this very promising technique?
Well, I think the first thing we have to do, I think that the whole stem cell field, in many ways, shouldn't be sort of lumped all together. I think the notion of hematopoietic stem cells, they are a huge advance. And they're going to continue to be utilized. In fact, they'll be utilized by these folks to do, I think, very important things. But I think that you have to dissociate real technology that has real near-term promise from, I think, where people went wrong with stem cells was this almost, lay notion that, oh, you can use stem cells and grow a heart or grow some organ. There's no science right now to get us anywhere near to that. So that was an example of overblown promise in the field that had no real basis in science. Okay? And the reason we're not growing hearts for people and putting them back in is not a safety issue, is they have no clue how to do that at all. There's no science to that right now. So a lot of the hype about, that entire field of growing organs using stem cells and all that was just totally overblown. I think for sure there are safety issues, as you guys addressed, and we'll turn them to, but it's a whole different thing in terms of there is obviously a real foundation in real science here where these guys are able right now to be going into mankind and have a very reasonable chance of success. So I'll turn it over to you guys to specifically respond.
A big part of our effort is devoted to safety. I mean, in safety in this case is specificity of cutting primarily, and looking at what the offcuts are, and then the risk benefit that John and Sandy were talking about. The diseases you choose need to be severe, and so the benefit of what you could do far outweighs the risk that you're going in with. But I think it's something that we're all concerned about. And I actually don't think the big problems are on this stage, quite frankly. I think the big problems are because the technology is so easy to use that there are going to be things that get advanced that don't have the industrial horsepower behind them to actually look at the safety issues and the specificity issues. So I do think we do have concerns in general about that. And we, our founders and others, and I'm sure for the other companies here, we are interacting with the regulators to help educate them about what a safe and specific gene editing product looks like.
Yeah. I'm sorry, that's exactly what I was referring to. I'm glad you stated that there are people who know what they're doing, but the problem in stem cells, as you quite rightly pointed out, there are a number of people who really know what they're doing, but a lot more who don't. And that actually damaged irreversibly, if you like, the field because there was so much hype. And my question then is, how do we prevent that from happening beyond the panel here and some other companies who really know what they're doing?
A couple of thoughts. We've had wonderful interactions with the FDA, much easier and productive than I had working on small molecules in bigger companies. They understand that they're part of this solution and have been very helpful. I don't know if you two have found the same thing. We do need to be very prudent, and one of the downsides of Sangamo is that we're the only company doing zinc fingers, or the really only company, and so we're the only person doing it. We know what we're doing. We've been going about it in a very professional manner because we own all the IP, because I believe IP is quite important. But
we own our IP as well.
But the fact that CRISPR is so easy to use, my worry isn't with the companies here because they really know what they're doing. It's that you can do it for $100 or less in your university somewhere, and what are the controls in that? And that's why I think a discussion about what good looks like has to happen soon, perhaps sponsored by all of us in a non-competitive way, so as we can guide society, we can guide the discussion of what we should all be aiming for.
Just one additional thought. I mean, yes. But I'm not worried about the companies. I'm not worried about the regulators for the reasons that have been stated. It is this kind of rogue thing where we find out someday that Dr. No on some island somewhere does something. We find out about it. And there is not a regulatory reaction to it, not an industrial reaction, but a public reaction to it. So that this notion of what everybody wants to do here and what we all want to address becomes very difficult to do because people fail to understand what the process is and what good science is and what good regulation is, because this one or two or rare instances of some spectacular thing catches the attention of the media and is what's talked about. I have a picture, the cover of The Economist, hanging in an office at work where it shows editing humanity. They've got a little baby. This is just last year, pointing to all of the parts of the baby's body that could be modified for enhancement of all kinds of things. It's exactly wrong. It's exactly wrong. It's not what we're attempting to do. And I think that kind of sensationalization really misleads the people in terms of what the medical problems are that we're working on.
After such impressive social thinking, I'm sorry to ask a dumb science question. There are lots of monogenetic diseases, or not lots, but a number, or copy number variants are the problem. Like Friedreich's ataxia, where the number of copy number variants is directly related to the severity of the disease. Can CRISPR or zinc fingers cut out the extra copy number variants? Because that's a gene you can't cut out. Do you follow? We can't just, like, knock it out.
Yes. Yes, I mean, you can make two cuts. You can actually introduce multiple guides or multiple zinc fingers into the cell and cut it multiple places in a single chromosome or multiple places in multiple chromosomes. As the distance between the cuts increases, the chances that they come back together becomes more challenging. Okay, so small deletions, very efficient right now. Very large deletions, the rejoining, it becomes a much less efficient process. So
that's an enormous copy number variant.
right. But sometimes you don't need, depending on the mechanism, you don't necessarily need to cut out the extra copies. We could go in and inactivate the extra copies. And you would need to identify genetic differences between the copies so you'd understand which copies you were inactivating, or if you wanted to inactivate all of them, obviously you'd find something in common.
Panel about societal reactions to the technology and its progress. How do we avoid an anti-GMO type reaction, which was huge and which has really destroyed the market in some sense for that technology? Do you have in your organizations an approach to winning the hearts and minds campaign?
We're 98 people right now. So it's a very small group. We're aware of those issues, though. And so, for example, in anticipation of some of these issues, the National Academy had at the end of last year a very public forum to address some of the questions that came somewhat with somatic therapy, but primarily with germline therapy. And we put a lot of effort into working with them to come up with what we thought was a responsible statement of principles. But getting people to understand that and promulgating it is a separate issue. We spent a fair amount of time in Washington talking to legislators who are on the front line to make sure they understand what the science is. We've been well received. People are intrigued. It's something new, and they try to understand it. And I think the early returns are positive. I think the test is going to be, though, when something goes wrong. And then we're going to find out, because will they hold the line, or will, or many people are, they're making a lot of noise, win the day. And frankly, it can't be us alone. I feel that the people who have the most at stake are the patients. In finding those affinity groups who have vested interests and understand what the technology, whatever form it takes, can bring to them. They are the allies that we need to have. And it's, for us, at least, it's very, very early days.
I also think it's very different. I mean, I think John alluded to before. If you start talking about enhancements, or you're starting to talk about more trivial things, maybe the ethical boundaries start coming into play. But what these guys are talking about is they're talking about blinding congenital diseases or hemophilias or these diseases which right now have no alternative. And I think there's no ethical questions about the value of doing a manipulation to save a patient's life, and it shouldn't really be any different whether it's a gene editing manipulation or some pharmacologic intervention. I think certainly if you go into other areas, and certainly if you go towards the germline and so forth, then ethical issues will definitely come up, and I don't know if we're prepared to deal with it, and I agree with John, there may be some crazy people who start doing it going rogue, but I think in terms of what these guys are doing, I don't think should be any sort of ethical question about whether it's warranted to save vision or to save a life. All right. Any more questions? All right, one more over there.
The genes that you edit, how many population doublings they go through, especially in the fastly dividing cells, like blood cells?
Once you edit, it's maintained through the progeny of that cell. So you're not going to lose the editors. For many of the subsequent daughters of that cell, they're going to be maintained. You're making a change to the genome. So it's not like a gene therapy where you have the potential, if it's not integrated, you have the potential to lose the change. This one will be maintained through the progeny of the editing that you've done.
Maybe one other thing to add to that, I guess, would be, you know, if you have a population of cells that slowly divides, but has as its progenitors some stem cell population, and you don't reach those stem cells, you could imagine, you know, based on whatever the doubling time is, and that's going to be tissue dependent, you could actually lose the edit. But that's, you know, that's going to be very much dependent on those cells that you hit and whether they're dividing or not.
Speak loudly.
So Dr. Macrae said this earlier, but I'll ask the question again. So you introduce a change, and it passes down through the progeny, every division, and so on and so forth. And then if and when something goes wrong, can you undo it? It's not withdrawing a small pharmaceutical, small chemical entity. And I think that's the thing that is scaring all of us, you know, is the permanence, potential permanence of this. I work in the diabetes care unit at Becton Dickinson. My name is Larry Hirsch. I'm an endocrinologist. And there's a huge interest in the diabetes field in stem cell-derived beta cell implantation as an ultimate quasi-cure of type 1 diabetes. And we know that the FDA has made it very clear that they are very, very concerned and likely will not approve going forward what we call microencapsulation, encapsulating individual cells in alginate essentially, because if something goes wrong, how do you pull them out? They want them in a macro device where if something happens, you can go in and retrieve the device. And I don't think it's not quite the same here, but in a way it is. I mean, so if you introduce gene changes, how do you undo it if something that you just could not foresee happens? And I think that's really what is the uneasiness about this.
So that's part of the discussion now, and that's why we should only be using these for important diseases where there's less alternatives. There are ways forward, so LMPs, you can repeatedly, theoretically, you can repeatedly dose, so you titrate up to a response and avoid overdosing. That's one. And there are some very clever molecular tricks that you can play where you put in a switch that would either activate the transgene or switch off the transgene when you give a small molecule. Although that means the patient's then got to take a small molecule, so you end up with a kind of one of these cell phones designed like a rotary phone. So I think we need to be very thoughtful about what diseases we attack with this technology. It is very powerful.
And I think what your question really gets at, I think Charles was addressing it early on. You're probably, for at least the early genes these guys are addressing, you're not going to want to reverse the change in the gene that they're addressing. It's really the off-target possibilities. You know, God forbid you've manipulated a second gene which makes the cell oncogenic, okay, that you don't even know about. So you're not talking about turning on or off the gene of interest or whatever. It's something that you don't know about. And these are going to be risks. I mean, you know, this is all about the technology. But it's all a matter of, is it worth the investment to give this an opportunity and try to figure out how to do it in the most responsible way possible? Because if you can figure it out, maybe you can be transformative. And with that, maybe I'll just take back the pulpit for a second. I mean, one thing which I think is very in line with the goal of the pre-Galien effort is to go back. The most important, I think, that we should all leave here, we should all go out there and remember that we got a big message to deliver out there to the world, okay? That, of course, there's risks, okay? But the biggest risk is not doing anything and not investing to try to change the world and save the world. And I think that we all have to be advocates for science and get that mission statement out there, that science is the only thing that can save the world, and that we are in dire straits. We really are. We cannot sugarcoat it, okay? Whether it's healthcare issues or environmental issues and so forth. And we desperately need science, and we desperately need to make huge investments. And we should be worrying about how to make those investments instead of trying to figure out simply how to contain the cost right now. It is about promoting science and innovation. So with that, we'll close the session, and thank you all for your attention.